Welcome back to biochemistry class. We come now to the second topic, which is about DNA transcription into RNA. Basically, we will talk here the process of transcription of DNA. Transcription is the first stage of the expressions of genes into proteins. In transcription, an mRNA or messenger RNA intermediate is transcribed from one of the strands of the DNA molecules. The RNA is called messenger RNA because it carries the message or genetic information from the DNA to the ribosomes where the information is used to make proteins. RNA and DNA use complementary coding where bases pairs match up similar to how the strands of DNA band to form a double helix. So RNA has the same nucleotides as DNA with one exception. And so much up to the split of the DNA. The exception is that RNA has adenine, guanine, cytosine, and guess what? It has uracil instead of thymine. One difference between the DNA and RNA is that RNA uses uracil in place of the thymine used in DNA. RNA polymerase mediates the manufacture of an RNA strand that complements the DNA strand. The RNA is synthesized in the 5 to 3 directions. So we are here now in the transcription part, as you can see, and we can see that there are the RNA polymerase that enters to the process where one strand of the replicated DNA are and will be transcribed by the complementary RNA bases. Mm, The main enzyme involved in transcription is the RNA polymerase, which uses a single-stranded DNA templates to synthesize a complementary strand of RNA. Specifically, RNA polymerase builds an RNA strand in the 5 to 3 direction, adding each new nucleotide to the 3 end of the strand. So, parang baliktaran lang sila. Yung template DNA is into 3 to 5 direction, while the RNA strand is in the 5 to 3 direction. The transcription process makes a complementary strand of mRNA in the nucleus. So, here is the process of transcription. We have here the initiation process. So the initiation of transcription in bacteria, or to be specific, begins with the binding of RNA polymerase to the promoter in DNA. Transcription initiation is more complex in eukaryotes, where a group of proteins called transcription factors mediates the binding of RNA polymerase and the initiation of transcription. Next process is the elongation from the word elongate or elongate. Mas parang may pinapahaba. One strand of the DNA serve as the template for RNA synthesis, but multiple rounds of transcription may occur so that make many copies of gene can be produced. Last of the process is the termination, from the word terminate. So it is the final step of transcription. Termination results in the release of the newly synthesized mRNA from the elongation complex in eukaryotes. The termination transcription involves cleavage to the transcript followed by a process called polydenylation. In polydielination, a series of adenine residues or poly A tail is added to the new 3 end of the messenger RNA strand. Let's tackle more about our let's go to the deep analysis in initiation process. RNA polymerase bind to a sequence of DNA called the promoter. Here you can see the promoter region found near the beginning of the gene. So each gene of groups of co-transcribed gene is in bacteria. So it has its own promoter. Once bound, RNA polymerase separates the DNA strands, providing the single-stranded template needed for transcription. Here in elongation, one strand of DNA, the template strand acts as a template for RNA polymerase. As it reads this template one base at a time, the polymerase builds an RNA molecule out of complementary nucleotides, making a change that grows from 5 to 3 direction. The RNA transcript carries the same information as the non-template coding strand of DNA, but it contains a base of uracil. So here in transcription, instead of thymine, we will use the nitrogen-based uracil. So here in the diagram or in the figure, we observe that the complementary bases of the template strand A is U or the uracil. Last is the termination part. Sequence called terminator signals that RNA transcript is complete. Once they are transcribed, they cause the transcript to be released from the RNA polymerase and from the RNA polymerase. An example of termination mechanism involving formation of hairpin in the RNA is shown below. So, makikita natin here in the figure na parang may hairpin structure. Nagkaroon ng parang hairpin structure yung RNA natin. And pag nakita na natin yun, it signifies that the termination process occurs.